Okay, ribosomes. Ribosomes are two subunits of ribosomal RNA and proteins, and they can be free in the cytosol, or they can be bound to the ER, which is the endoplasmic reticulum. Ribosomes, they synthesize proteins. Their function is in protein production, and free ribosomes are suspended in the cytosol, meaning they're not really bound to anything. They're just like in the cytosol doing what they have to do. They synthesize proteins that function within the cytosol, and bound ribosomes, they're attached to the endoplasmic reticulum. They synthesize proteins for export membranes. And prokaryotes and eukaryotes have different ribosomes for whatever reason. The ER, the endoplasmic reticulum, it is an extensive network of membrane-bounded tubules and sacs, and these membranes separate lumen from cytosol when they're continuous with the nuclear envelope, which is the double membrane that I talked about earlier, with the nucleus and its double membrane and its permeability and stuff. The endoplasmic reticulum's function is it manufactures membranes and performs many biosynthesis functions. And this basic structure of the endoplasmic reticulum, it's a membrane connected to the nuclear envelope, and it extends throughout the cell. So it's kind of like you see the big circular nucleus, if it really is circular. I don't know, I can't see that small. And then you have like these little, use your imagination. There's two types of ER, which is smooth ER and rough ER. Smooth ER, it's the factory processing operations. It's got many metabolic processes, and it's synthesis and hydrolysis, which is the breaking down and building up. And then enzymes of smooth ER, which it synthesizes lipids, oils, phospholipids, steroids, and sex hormones, which we produce. And the hydrolysis of glycogen in the liver into glucose, and it detoxifies drugs and poisons in the liver once again. Like alcohol and stuff. Rough ER, sorry, got distracted. Smooth ER and rough ER. And rough ER, it produces proteins for export out of the cell. Like, so you have the smooth ER and it's doing all this stuff for inside of the cell. Then you have the rough ER and it's doing stuff like it's taking what the cell has made and it's sending it out. Rough ER, it produces proteins for the export cells. It's protein secreting cells. I said secreting, right? It's packaged into transport vesicles for export. I'll tell you what vesicles are later. And it's basically a membrane factory. Golgi, whatever. That apparatus. It's basically a stack of flattened membranes, flat membranous sacs, as the book says. And it has polarity, which that makes it end up being two different kinds. One of them is like the cis. I don't know if you're supposed to say CIS or cis, and then there's the trans, and I'm gonna say trans because it sounds like trans. And it's the cis and trans faces. The flat membranous sacs are called cisternae, and it supposedly looks like pita bread, but I've never seen pita bread in my life, and if it looked like that, I probably wouldn't eat it. And it has its two sides. Structure determines function, so therefore the different structures of the Golgi apparatus ends up having the different function, the cis being one of them. It receives materials by fusing with vesicles that's receiving. And then the trans is, it buds off vesicles that travel to other sites, which is the shipping or the transfer. Golgi processing. During the path from cis to trans, products from the ER are modified into their final form. Tags, sorts, and packages into transport vesicles. Something else it does. And the Golgi is like the UPS headquarters of the cells, Yay. And the transport vesicles are like the UPS trucks that actually go out, that the headquarters are the one who's like, okay, this is the ones that need to go there, but the trucks are the actual things that get them there. And that's it for that one.